Welcome, welcome, welcome. It's Mr. W from learn-biology.com, and I am so happy to welcome you to the first of 23 live review sessions that I'm going to do between now and the AP Bio exam. We're going to go all the way to May 15th. The exam's on the 16th, and I'm committed to you, and I'm making this public commitment right here. 23 live review sessions. We're going to start by finishing the course, since that's what most people are doing. So I'll be doing live review sessions about evolution and then ecology. And then really it's up to you. It's going to be based on the comments, the emails that I receive. If there's a topic that you're finding that's particularly difficult, that's what I want to teach about. So it's so great to have you here. And it's also so great to have you here teaching about this incredible stuff, which is evolution, which makes me so happy to teach about. Let's go. So here's what we're going to cover over the next couple of sessions. Here's the material that we'll be covering in Unit 7. So we're going to start by talking about selection, natural selection, artificial selection, sexual selection. We're going to take evolution all the way back to Darwin, who developed these phenomenally important ideas. Then we'll talk about population genetics and Hardy Weinberg. That's kind of genetics that was discovered about 100 years ago or so, and is one of the most important ways of thinking about evolutionary change. We'll look at the evidence for evolution. We'll look at phylogeny, the branching structure of evolution, which is really what evolution is all about. We'll talk about the incredible processes of speciation, the variation that creates variation, the variation that creates speciation, and we'll talk about extinction. And then we'll talk about the origin of life. So this is phenomenally cool stuff. I love teaching about it, and I'm so happy that I get to do it with you tonight. My name is Glenn Wolkenfeld, also known as Mr. W. I'm a retired AP biology teacher. When I taught in the classroom, most of my students got 4s and 5s on the AP Bio exam. I'm pretty confident that with this series of videos that I've been putting online for about the past month and a half, and the materials that I've created you on my website, you can get there too. And in fact, I provide a money-back guarantee for people who go through learn-biology.com. And I'm just realizing right now that I have to make a little change. Excuse me for a sec. So I'm going to go like this, and we're going to go here. Sorry about this. We're going to go slide. We're going to go edit theme, and this box has got to go. Yeah, box is going to go up here. Now we're just going to go right back to here. So where was I? I think where I was I was telling you that for folks who finish learn-biology.com, which is an interactive online AP biology curriculum, we provide a money-back guarantee. If you don't get a 4 or a 5, we give you your money back. You're starting a little late if you're starting right now. But if you had started in September, you could easily have mastered all this material. And who knows, maybe you're going to pour your heart and your soul into it. There's a couple of days left in a free trial that we provide. We provide a free trial from June 1st to April 15th. We have to turn that off on April 16th. But even if you start on April 16th, 17th, 18th, a couple of weeks that you spend with learn-biology.com will be transformative to your understanding of biology. A subscription also comes with access to the Biomania AP Bio App. It's the best app for learning biology. Both learn-biology.com and Biomania have interactive multiple choice questions that give you feedback. They have click-on games. They have flashcards. They have interactive FRQs. And in addition, learn-biology.com is a complete interactive textbook. You can take your textbook and get rid of it and use Learn-Biology instead. It's 1,000 times more effective. But let's get into what we're going to talk about right now, which is evolution. As you get ready for the AP Bio exam, you are going to need a checklist. You're going to need to identify what material you know well and what material you don't. You can do that using the College Board's course and exam description, which is over here, but you will be much more successful if you download our AP Bio Study checklist, which is a checklist that's got little checks that you can check off so you can identify the material that you know well and the material that you need to study. I actually teach about a method that's called the stoplight method that will help you do that. Anyhow, go to apbiosuccess.com/checklist, and that's where you'll get our AP Bio checklist. And again, that's just one of the many resources that we're providing to you at learn-biology.com so that you succeed in the AP Bio exam. So we're just going to cover 2 topics tonight. I'm not quite sure how long that's going to take. And the 2 topics that we're going to cover are natural, artificial, and sexual selection, really the bedrock of understanding evolution and evolutionary biology. And then we'll get into Hardy Weinberg and population genetics. Let's do it. So here are the questions that we're going to be addressing. And as we do that, I should probably set myself over here. And the questions that we'll be addressing are, what is artificial selection? Explain natural selection. And what is sexual selection? So again, these are all ideas that were developed by Charles Darwin in the 1830s, 40s, 50s. He encapsulated all these ideas in his fantastic book, The Origin of Species, which you should put on your must-read list, but not now, because right now you're probably an AP Bio student who is here that's getting ready for the AP Bio exam. I want to emphasize something before I get a little bit more deeply into this material, which is, I see there's a bunch of people watching, and what I want you to do is this: feel free at any time to put questions into the chat. And in fact, what would make me feel really happy right now is if you just said something like, "Hey, Mr. W, here from wherever you are. I'm broadcasting this from Berkeley, California, so I'm very curious about knowing where you are." And I'll probably wait to answer questions. So if you put a question in the chat and I don't answer it right away, I want you to know that inside I'm just ecstatic that there are people who are watching this, who are asking questions. But just to keep the flow of the presentation going, I'll probably wait until there's some kind of natural break point in the presentation, and then I'll answer your questions. So please ask, please stay, hang on. I promise that I'll get to every question. And if we run out of time and I can't answer it tonight, I promise I'll answer it. I'll answer it in the next live, or if you leave me a comment on YouTube, I'll send you an email. If you leave me your email address, people don't usually do that, I'll answer in the comments. So that's how it goes. So again, I want to encourage the people who are watching tonight, leave me comments. And again, just like post a comment right now, just say, "Hey, Mr. W, how you doing? I'm here from wherever you are." So I'd love to know who you are. Please leave me comments. So now that we talked about the questions that we're going to address in this first section, let's do it. So we're going to start off by talking about what artificial selection is. And here I have a picture of a bunch of common vegetables. And the astounding thing is that all of these vegetables are varieties of the same species. That species is Brassica oleracea. That's a common mustard over here. That's what it looks like. And through artificial selection, it's been bred into distinct varieties that are all the same species. And sometimes you see hybrids between the two of them, like broccoli and stuff like that. So cauliflower, broccoli, Brussels sprouts, kale, kohlrabi, and cabbage have all been bred from this plant over here. How is that done? Understanding this was something that fascinated Darwin, and he used this as an analogy for understanding natural selection. So artificial selection is merely selective breeding. And the way that it works is that breeders envision a desired trait in an animal or a plant. So for example, they had this idea, some breeders had this idea, well, let's breed for the flower clusters because they create a nice kind of meaty thing that you can put into the oven and broil up, and that's fantastic. Or let's breed for the flower buds, and let's breed for the lateral buds. That's how you get Brussels sprouts, or the leaves or kale, or the stem is kohlrabi, or the terminal buds or our cabbage. And you have to do that for many generations. So you select organisms with that trait. And what that would mean is, for example, in the cauliflower or the broccoli, you'd collect seeds from plants that have desired characteristics, and you'd only plant those. So in every generation, you're selecting the organisms that have the traits that you want, and you are therefore creating what is a carefully guarded gene pool that only includes organisms within the population with genes for the desired trait. You do that for a couple of hundred or thousands of years, and that's how you create these different varieties. And of course, it can happen much faster with modern technologies. I don't have a picture of dogs, but all dogs are the same species as wolves. They're all varieties of Canis lupus, but the different varieties of dogs have been bred for different behaviors, different characteristics, so on and so forth. So that's artificial selection. Now, artificial selection was Darwin's basis for his theory of natural selection. He developed that over a long set of decades that happened in the 1800s, culminating in his publication of The Origin of Species in 1859. So here we see a kind of reptile that's called the satanic leaf gecko. And the satanic leaf gecko looks weirdly like a stick or a leaf. It's beautifully camouflaged. Now, nobody went ahead and bred the satanic leaf gecko. So how did it develop this form? How did it develop this adaptation? An adaptation is a trait that promotes survival and or reproduction. So here's how it works. In every population, there's variation that comes about from processes that you've learned about earlier in your AP biology education: genetic combination and ultimately mutation. Mutation is what's introducing new alleles into any population. The kind of variation, little typo here, the kind of variation that matters for natural selection is caused by heredity. So you can generate variation by different environmental regimes, different environments simply, and that's how you can create one person who might be skinnier and one person who might be heavier by exposing one to more food and the other to less food. But the kind of variation that we're talking about here is genetic variation that's caused by genes. Now, in every species, the parents produce offspring at a rate that exceeds the environment's ability to support them. In other words, many are born, but few survive. Now, that's of course true of organisms like bacteria, which can reproduce themselves every 30 minutes. But even slow breeders like elephants will produce more offspring than can survive. And that is simply a law of nature. Species that don't do that wind up becoming extinct. So all the species that we observe that are doing okay are ones that are reproducing at that level. And overproduction of offspring means that there's differential survival. Not everybody survives. So too many elephants are produced. Which one gets to survive in the context of there being a finite world with finite resources? Well, it's the ones that have beneficial traits. And if you take that process and you continue to add mutation, which creates new variants, and then you repeat that cycle over and over again, you wind up with remarkable adaptations. Think about the wings of a bat, which have become adapted from, this kind of same structure. But years and years and really generation after generation of selection led that structure to develop into a wing. Think of the bat's ears. In the same way, the bat's ability to detect its own echoes, its ability to echolocate, which is the same thing that dolphins do, that came about through year after year, generation after generation of selection in the same direction. So that's how natural selection works. It's the most important idea in biology. And I see that. So unfortunately, I put myself over on the side over here. That would have been a better placement, or maybe even over here. Got to be a little bit more aware of that. Sorry about that. So anyhow, that is the story with natural selection. I'm not seeing any questions come in, though. I'm seeing a healthy number of people in the audience, and I actually want to encourage you again. Now would be a great time to throw any questions in. But let's go on. Also, what would be super useful would be like an audio check. Like if somebody were just to chime in, "Hey, Mr. W, hearing you're okay," that would be a sort of reinforcing gesture that would make me more confident. So if one of you people who are listening could just put in the comments, "Got it, Mr. W, we hear you okay," that would be super useful. And adaptations arise from repeated selection in the same direction. So in the case of this satanic leaf gecko, there's always going to be predators who want to eat this gecko, or on the other hand, there might be prey. So the better that the gecko is camouflaged, the more successful it would be at avoiding predators, finding its own prey, and that's how we move evolution forward. Adaptive melanism is an idea that comes up a lot in AP biology. And what's frequently done in AP biology is you frequently are involved in activities from HHMI, the Howard Hughes Medical Institute, and they have fantastic data sets. And those data sets often appear on the AP bio exam. So what is adaptive melanism? Need a moment to just take a sip. If you're looking at this diagram and you're having difficulty seeing the mouse over here or the mouse over here, that's exactly the point. Adaptive melanism is the darkening of fur, scales, feathers, or other appendages, wings in insects, for example, within a population in response to the darkening of the environment. And that's great and good. I just saw a comment from Sophie, and despite my bravado earlier where I claimed that I wouldn't stop whatever I was doing, I become so excited whenever anybody actually comes on board and leaves me a comment that I just want to thank you very much, Sophie. And I'm seeing a bunch more people. So if other people might want to chime in about like who you are, where you're coming from, and that's fantastic, Sophie. I'm really hoping that this is going to be useful. I'm going to do another review. Let's see, Thursday, that ends this week. I'll tell you about my schedule of reviews, and maybe I'll throw it in Sunday night for you. So adaptive melanism, I just want to emphasize, like today I went swimming. My skin darkened on account of the fact that I went swimming because I tanned pretty nicely. That's not adaptive melanism. That's an immediate adaptation to an environmental pressure, and that actually protects my skin from ultraviolet radiation. There's many good things, but that's not what we're talking here. What we're talking about is a population level change, where the population darkens over time. So in other words, from a common ancestor, we have an evolutionary change where this population of mice actually adapted to become dark. Why? Because in the deserts where these mice live, this is the rock pocket mouse, and there's a fantastic HHMI about this. These mice migrated onto lava flows. Lava flows are dark. In that environment, there was tremendous selective pressure. What do I mean by that? I mean hawks would just eat these mice. So any mouse that didn't blend in with the environment would wind up being eaten. Its genes would literally wind up being removed from the gene pool. A mutation emerged in some of these mice that led to the darker coloring. And I believe we'll talk about that a little bit later, either tonight or another night. And that selective pressure that drives adaptive melanism is typically predation. And just like I was talking about, hawks, owls, other aerial predators, any mouse that didn't blend in with the substrate, with the dark rocks that it was living on, would literally be a hawk snack. So that's really what this is all about. And I'll talk about this graph in a moment. All right, list the 3 ways in which natural selection can change the distribution of phenotypes in a population. I'm going to move myself again. How's that? No. That's good. Okay. So now I'm out of trouble. I guess I could put myself over here for right now, but not really necessary. I'm wasting everybody's time. You really didn't need that, nor did you need that. You're probably thinking, this guy doesn't know what he's doing. But here we are. I'm safely on the side. So list the 3 ways in which natural selection can change the distribution of phenotypes in a population. So let's talk about that. So in any population, most characteristics can be represented by a bell curve of continuous variation. In other words, ignore the red graph, the red line over here, and just think about the black one. So if this represented, for example, height of all of the students at your school, there'd be some who are short, there are some who are tall, and then most people cluster around the middle. Most genetically determined traits are like that because they're caused by a multiple array of genes. Now, you start from that. If you're measuring body length, most individuals are average length with fewer that are shorter, fewer that are longer. In directional selection, which is shown here in graph A, you're selecting against one of the extremes. So like, for example, in adaptive melanism, if the value of the trait was amount of pigment that you produced in your fur, then you're selecting against the light color over here, and you're selecting for the dark color. And what do? You shift the population over to the right. And what happens is that because there's renewed mutation, renewed recombination that happens in every generation, you might temporarily change the shape of the curve, but later it widens out again so that in any population, there'll be an average amount of darkness, and some will be really dark and some will be a little bit lighter. And the hawks will keep on selecting these ones over here, driving it over to the right until the population is adapted. So that's directional selection. This is called stabilizing selection, and that's selection against both extremes. An example of that one is really common to our species, which is birth weight in babies. There's selection against very high birth weight babies because they have difficulty leaving the mom and being born. And in fact, there can be complications that can kill the baby, kill the mom during birth. And that was much more common before there was modern obstetrics and modern technology that was associated with birthing. It's also not good to be a low birth weight baby. So there's selection against small size, there's selection against large size. Most babies wind up being somewhere in the middle. This is much harder to find examples of and think about, but when we talk about speciation, we'll come back to it. So that is disruptive selection, and disruptive selection is selection against the mean. And what you wind up doing is there's a situation in which the average trait is somehow less adaptive, and then you wind up selecting for the 2 extremes. So those are the 3 kinds of selection: directional, stabilizing, disruptive. Any questions about that? Please put it in the chat. Just do like what Sophie did and let me know what you're thinking and what you need. That leads to this idea of evolutionary fitness. We live in a fitness crazed culture. So people tend to mistaken fitness with strength, but it's not that at all. Fitness is simply the number of offspring and offspring of offspring that survive to reproduce. And I love this diagram of penguins, the emperor penguins, and their life cycle, because it shows how many dimensions there are to penguins. So, how many dimensions there are to feeding. So one dimension to fitness is how well you swim. Another dimension is how well you can walk to the rookery. Another dimension occurs during the mating process. That also involves sexual selection, so we'll talk about that in a little bit. But then it's how good you are at incubating eggs, and then how good you are at taking care of the young. So it's so many different kinds of dimensions that are involved in fitness. And all of that together leads genes that get passed on from one generation to the next. So again, never think of fitness as anything as simple as speed or strength or anything like that. Fitness is about your ability to create offspring that themselves can survive to produce offspring. It's a complex ensemble of traits. Reproductive success is what fitness is all about. As a great example of natural selection and fitness, let's look at this example. Explain how the peppered moth demonstrates an evolutionary change in response to an environmental change. So the peppered moth is an interesting example because it is one that was observed in historical times. Shortly after Darwin published The Origin of Species, remember that's 1859, there started to be observations by natural historians, early biologists, who were actually observing this kind of change. So what happened? The natural form of this moth, Biston bitularia, is this over here. It's originally, the key point is that there's directly observed directional selection and adaptive melanism. So here's the natural form of the moth. Its predominant phenotype was peppered. In other words, if you think about what you'd see if you just took the pepper shaker and put it on a white table, you'd see something like this. You'd see, mostly white with black specks. The reason for that was that was fantastic camouflage for these moths as they rested on trees, and it protected them from their key predators, which are birds. So here's the form that predominated in places like northern England before the Industrial Revolution. And remember, what we're talking about is stuff that started to happen in the 1820s as we shift over from an agricultural society to an industrial society. There's massive mining of coal, and that coal is being used to power factories, and those factories are pouring soot and smoke into the air. So what happened is this. If you look at the beginning, there are very, very few black moths, and the predominant phenotype, that's a word from genetics, the appearance, is mostly white. But as the Industrial Revolution progresses, what happens is that the mean phenotype shifts from peppered to dark. Why is that? Because of camouflage. So basically, the situation that the moths were living in changed so that the dark moths were suddenly better camouflaged, and the white moths were simply sitting, I was going to say ducks, but really they're sitting moths. So here's what the situation looked like after industrial melanism. Now, the really cool thing is that starting in about 1960, there was worldwide pollution controls that were instituted around factories. So the soot from the factories went down, and all of a sudden the trees, instead of looking like this, started looking like this again. So once again, what would have happened had this graph continued would be another decline in the number of black moths and the rise in the number of white moths as the trees started looking like this and like this. So we have moths shifting from light colored to dark and then back to light. And again, it's not individual moths that are shifting. Individual moths are simply surviving and reproducing, or they're being eaten. And when they're being eaten, their genes are removed from the population. So that's the situation with the peppered moth. It's a great example of natural selection that's been observed in modern times. In case you're wondering, I have a Science Music Videos mug. It's available through, right below the video if you want to get it. And isn't that fun? Okay. We've talked about artificial selection. That was our example with broccoli. I also mentioned dogs and things like that. Then we talked about natural selection, how the process worked. We talked about types of selection. We talked about adaptive melanism. And we had this illustrative example of the peppered moth. And now what we're going to talk about is another process that was also identified by Darwin. It's called sexual selection. And the definition is selection for traits that directly include reproductive success. Often these traits are deleterious. Often these traits are negative in terms of natural selection. So like, for example, think about this bright coloration that forms the wattles of these turkeys. Or think about their tail fans. Their tail fans don't really assist them in flying. And the ultimate example of this, of course, is the peacock, who, has a lot of difficulty if you're a male peacock getting off the ground. So these are characteristics that often decrease fitness in one sense, but they really increase fitness in another sense. And the thing that I love about this picture is I took it in a walk around my neighborhood here in the Bay Area of California. And there were a bunch of male turkeys that were displaying for a female turkey. And notice these characteristics, these incredible fantails, the size, the wattles that are on the males and on the females. So what happens is members of one sex in this form of sexual selection that's called intersexual. That inter, like international, is trade between nations, international trade. Intersexual selection is selection that happens between the sexes. So members of one sex, typically the females, but not always, choose mates of the other sex, and those are typically males. And the result is colorful, highly adorned males and choosy females. So there's a dynamic that's caused by intersexual selection where the males develop these traits. They pass on the genes for those traits if they're successful at mating with the females, and the females pass on their preference for those traits. So it becomes a kind of positive feedback loop that leads to extraordinary things like what you're seeing here, or just imagine the tails of a peacock. There's another and quite different form of selection that's called intrasexual selection. And that is one where there's competition between males for control of a harem of females or control of the breeding territory. This is not a picture that I took myself, but I have seen this myself because close to where I live is a nature reserve called Año Nuevo. And at Año Nuevo, New Year, in Spanish, there is a colony of elephant seals. And those elephant seals are highly territorial insofar as the males will control the areas where the females come to shore to give birth. And then after giving birth, the males mate with the females. And the males are like 3 times the size of the females. Why is that? Because there's competition between the males to control these breeding areas. There's not similar competition between the females. So females are, big, and the males are huge. And you think about animals where there's a big size difference like that, like for example, among our close relatives, gorillas, because of sexual selection where males control a harem of females, the dominant male is gigantic. The females are much, much smaller. So that's sexual selection. And it results in sexual dimorphism, different phenotypes in males and females. We've talked about intersexual selection and intrasexual selection. What I want to do now is just quickly pause and just tell you a little bit about my website. And I'm just going to go like this in order to make that happen. At learn-biology.com, we understand why students struggle with AP Bio. It's a hard course, but we have a plan for your success. Go to learn-biology.com, sign up for a free trial, and complete our interactive tutorials and interactive AP Bio exam reviews. We guarantee you a 4 or a 5 on the AP Bio exam. See you on learn-biology.com. At learn-biology. So learn-biology.com, I'll tell you a little bit more about later. I'll even show you, but it has quizzes, flashcards, interactive diagrams. It has complete tutorials. It's got fantastic AP Bio reviews. So I want to encourage you to sign up while you still have a couple of days left to do this for a free trial. Just head on over there, sign up for a free trial. And where you want to go to is learn-biology.com/student free trial. I'll show that at the end. Give it a try. You'll learn so much. You'll realize that this is the tool that you want to have as you prepare for the AP Bio exam. Let's move on to our second topic of the night, and that's population genetics and Hardy Weinberg. Here are some of the questions that we'll be addressing in this part of the video, and those questions will be: describe the... I didn't update these questions, and that's so unfortunate. But what we're going to do is we're going to talk about some really cool stuff. We're going to talk about what is population genetics, what kind of analysis do you do in population genetics, what is the evolutionary approach to population genetics. In other words, when you use a population genetics lens, what kind of evolutionary biology can you learn? And finally, there's a whole mathematical aspect to population genetics that is actually fairly uncommon within our biology course, but here it gets pretty intense, and I'm going to walk you through some techniques for learning about the mathematical parts of biology. So to begin with, what is population genetics? It's the study of how genes are distributed in populations and how they change over time. The key measure is allele frequency. So here we have a gene pool of 20 individuals. It couldn't be simpler. Every human being has 20,000 genes. There's a similar number in a mouse or in a nematode worm for that matter. But here we're just looking at one pair of alleles, one gene that's controlled that we're going to call A. Maybe it controls color or something like that. And basically, what we see here is the distribution of big A and little a in 20 mice. And what you can do is simply count up all of the A's in each row. So like 1, 2, 3, 4, 5. So there's 5 little A's in this row and 4 in this, 4 in this, 7 in this. If you add that all up together, then what you get is 9 plus 11. So there are 20 out of 40 alleles. So what's the frequency of little a? 0.5. We just did some population genetic analysis. It's that straightforward. It's that simple. So how do we think about evolution in terms of genes and gene pools? Well, first of all, let's define the term gene pool. The gene pool is all the alleles of all the genes in a population. And evolution, when you look at it in terms of gene pools, is a change in the genetic makeup of a population over time. So what are we talking about? You know, if we talk about this gene pool, which consists of genes A through E, it would be the frequency of big A, the frequency of little a, the frequency of big D, little d, big E, little e, and how those change over time within that population. So what kind of changes are we going to be seeing? We're going to see changes that are adaptive, that result from natural selection of phenotypes that are associated with specific alleles. And that's actually a very important idea. Natural selection doesn't select genotypes, it selects phenotypes. And then what it's essentially doing is it's increasing the distribution of alleles in a population that code for favorable phenotypes and decreasing the distribution of alleles that code for harmful phenotypes. So the changes can be adaptive. The changes can also be negative in terms of random changes that decrease the fitness of a population, and they can be neutral as well. So let's now get rid of the biggest misconception in population genetics. And that misconception is this idea. I tried to represent it in a way where it was clear that it's a misconception. So this is a wrong idea. And I heard it from students throughout my teaching career who would ask me questions like, Mr. W, Shouldn't there be more of the dominant allele than the recessive allele? And the answer to that is no, no, no. That's a wrong idea. The truth is that the frequency of an allele is based on the advantage, the benefit, or the harm that that allele confers to its associated phenotype, or random historical factors. And just to give you a very human example, roughly speaking, blue eyes, blonde hair are, oh, thank you, sometimes X Ramen, for your comment. See, I get distracted by these comments. I like to think that I'm cool enough to have people wait, but really I'm just so thrilled whenever anybody comments. So basically, if you think about the phenotype of having blue eyes and blonde hair, these phenotypes are actually complex, and they're actually controlled by multiple of genes, but very loosely speaking, blue eyes is recessive and darker colored eyes are dominant. We know that there are other shades of eyes, and there's hazel and all those things too, but we're not worrying about that because we're just doing a little thought example. And the idea is that if you go to someplace like Norway or Sweden, most of the folks who live up there have blue eyes, and that's a recessive allele. Why? Well, it's actually about natural selection. There's been selection for less melanin so that people who live in northern climates will be more effective at creating vitamin D. And there's actually a less of a need for protection against ultraviolet radiation. So dark colored skin is an adaptation for ultraviolet radiation protection, and also for the protection of, it keeps the vitamin, which is folic acid, from being destroyed. Anyhow, I went a little bit off the rails there, but it's totally possible for a recessive allele to be very high in frequency. Dominant alleles are not more common. So we've got that misconception out of the way. Hopefully you won't have it. And the reason why I have this picture of Kyle Paycheck, as I believe his last name, Pasek, is he has the genetic condition achondroplasia. It's a form of dwarfism. And basically that's a dominant allele that causes that. But you don't see a lot of people with achondroplasia on the streets because in some ways it's a negative phenotype that has other health impacts. So dominant allele, very rare. There's no connection between those things. And that was what I was just saying. So let's talk about genetic drift and how a population bottleneck works. So genetic drift is a condition where there's a random change in allele frequencies that's usually associated with small population size. So what I've represented here is a bunch of pigs in a population, and they have two phenotypes, dark and light colored. The light colored requires the recessive, two recessive alleles. Now, if there were some situation where randomly, none of the black colored pigs were able to pass their genes on, then what you'd have is essentially elimination of an allele from a population. So a population genetic bottleneck is where there's some biological or abiotic, some kind of something in the environment that's not connected to biology. It's a term we'll look at a lot in Unit 8, that wipes out a large percentage of individuals in a population, leaving only a few survivors. And the survivors' alleles might not be representative of the allele frequencies in the former population. And in that former population, there was relatively high representation of the big B allele, but now in this generation, it's fallen to 0. That's a population bottleneck, and it can drastically reduce genetic diversity. And again, what I want to emphasize is that the survival of the little b allele, this isn't natural selection. This is just sort of a random change. Like, for example, a good way to imagine it is that there was a virus that wiped out a large portion of the individuals in this population, and the virus randomly wiped people out. But when that viral epidemic had passed, the only survivors happened to be these two white pigs, and they passed their genes on. The reason why it's called genetic drift is that when you drift, you're not moving with intention. You're sort of letting the current take you, and that's different from selection, which is a very active process. So this is much more passive. It's much more random. So that's genetic drift, population bottleneck. And the overall effect is that new allele frequencies in the surviving population might be quite different from those in the parent population. A related phenomenon is called the founder effect. And the founder effect is another type of genetic drift. And it can occur when a small number of individuals from a large population found a new population. Now, I express this in a kind of statistical way, but basically it's because of insufficient sampling. The allele frequencies in the gene pool of the founders might be different from those in the parent population. So let's look at some examples. Here's the parent population, and they're sending out founders. Now, what do I mean by this? These might be birds who are colonizing islands, or they might be human beings who are colonizing islands. So the red squares and the blue circles represent alleles in a gene pool. The top and the bottom populations have lost an allele. Why? Because the founders that happened to found the population on this island population, they all had the red allele. There was nothing that was adaptive. I need to emphasize this over and over again. It's not that the red allele was superior. It's just the folks who left on the boat just all had this red allele. And in this case, all the folks who left had the blue allele. So here we have populations that have lost genetic diversity. And in the middle population, the blue allele is in higher frequency than it was in the parent population. So again, this is called the founder effect. It's a form of genetic drift. In that way, it's related to a population bottleneck. The example that you probably learned about in your biology class is the Pennsylvania Amish, who were founded by a small number of people who came from Europe. And they have a high frequency of a genetic condition that's called Ellis Van Creveld syndrome. Why? Because the founders of the community brought that allele with them, and they were sort of oversampled in the subsequent population. Another phenomenon that can change allele frequencies is gene flow. Gene flow works like this. It's the movement of alleles from one population to another. And this can involve the movement of individuals who bring their alleles with them, or the movement of gametes, such as pollen, or carried by the wind, or being carried by pollinators. So over here, we have a population that's mostly brown. Over here, we have a population that was mostly black. And basically, individuals from the brown population are now intermixing with this. So what are we doing? We're not really changing the allele frequencies in population 2, but we are changing them in population 1. Why? Because of gene flow. In either case, the effect of gene flow is to diminish the differentiation between adjacent populations. What's the effect of mutation upon evolution? Two things. Mutation is the ultimate source of genetic variation within and between populations. And if there is a persistent change, like if, for example, this nucleotide C tended to regularly change, that's a bad example. If this T over here regularly changed to C, then what we would be doing is we'd shift the population in one direction. In other words, this phenotype that's associated with this basic amino acid would increase in frequency. And again, remember, why are mutations important? Because they're the basis of the phenotypic variation upon which natural selection can act. What's the Hardy Weinberg principle? In your response, include the 5 features of a population that's in Hardy Weinberg equilibrium. Hardy Weinberg describes an idealized non-evolving population. This idea was developed in the 1900s, and the situation is one that actually doesn't exist, but it's used as a foil to explain how natural selection, how other factors can change allele frequencies in various populations. So this fictional population, idealized population, has 5 features. And if any of these are not adhered to, then allele frequencies in that population will change. So one of those is that the population needs to be infinitely large. Really, that means it has to be big, because if it's small, then there can be genetic drift, which we've seen as a function of population bottlenecks or population bottlenecks or founder effect. No allele can be harmful or beneficial, because if an allele codes for a phenotype, produces a phenotype that's harmful or beneficial, then that allele will rise or fall in the population. And you're probably saying, well, how could that be? It can't be. The purpose of Hardy Weinberg is to present this idealized case in which no evolution is occurring. If an allele codes for a beneficial trait, then that allele will rise in frequency. If it codes for a harmful trait, it'll decrease in frequency. Random mating. There can't be any sexual selection. So every phenotype needs to be equally attractive, and that will keep allele frequencies the same. There's no emigration or immigration. No alleles are entering the population, none are leaving the population, so there's no gene flow. And finally, what we just discussed, there's no net mutation of one allele to another, which would decrease the frequency of the first allele and increase that of the second allele. So now that we know about the Hardy Weinberg, we can actually look at some mathematical models for how population change can be measured. This is the Hardy Weinberg equation, p squared plus 2pq plus q equals 1. What is this equation about, and what does it enable you to figure out about a population structure? So what this means is that the frequency of homozygous dominance, p squared, why? Because in this model, p represents the frequency of the dominant allele, and q represents the frequency of the recessive allele. So if you are p little p squared, then you'd be homozygous dominant, right? Well, if you're PQ, then you're a heterozygote. You have one dominant allele, you have one recessive allele. And if, as you can see in this Punnett square, there have to be 2 of those. And then if you're Q squared, then you are homozygous recessive. All of those individuals together for that one allele represents 100% of the individuals in a population. Now, of course, this is only true in a population in which there are genes with 2 alleles. If you have 3 alleles, you can still do this. So like, for example, if you think about the ABO blood type system, you can do Hardy-Weinberg. It's just more complex, and that will not happen to you at the AP Bio level. So all you have to worry about is genes which have 2 alleles. And once you know the frequency of the recessives, Q squared, then you can figure out the frequency of everything else. Now, there are those of you who are mathematically gifted, and you can go ahead and you might be able to whip out your scientific calculator and figure this out, but I recommend that you use a cross multiplication table. I'm going to demonstrate that right now. So here's a sample problem. You've identified that 49% of the mice in a population have the recessive trait of white fur. Figure out the frequency of the dominant allele, P, the recessive allele, Q, the number of individuals who are homozygous dominant, the heterozygotes, 2pq, and Q squared. So Q squared is 49. How do you know that? Because if you have 2 copies of the recessive allele, then you're going to have the recessive phenotype. So a trick to knowing all these problems is that the number that they usually give you is Q squared. So what that means is this: if Q squared is 49, that's given in the problem, then the square root of 0.49 is 0.7. So what we're going to do is we're going to load 0.7 over here, representing Q, and over here, representing Q. P, Q, P, Q. Now, if this is 0.7, then the frequency of the dominant allele has to be 1 minus 0.7, which is 0.3. Again, this is a two allele system. So if one allele has 0.7 frequency, then the other allele has to be 1 minus 0.7, which is 0.3. Or if you think about this, it might be a little easier. Think of this as 70% of the alleles are little a, then 30% have to be big A. But it'll work better if you don't work with percentages, work with decimals. Now, once you have that figured out, then you can figure out that the number of heterozygotes is 0.42. How do you do that? 0.3 times 0.7 is 0.21. 0.3 times 0.7 is 0.21. Those together constitute the number of heterozygotes. So that's 0.2. P squared is 0.09. 0.3 times 0.3, 0.09. Less than 1% of the individuals in this population have that genotype. In this case, it would be big A, big A. And then often what's thrown in for fun is, well, what percentage of the individuals would have the dominant phenotype? Well, then you just have to add this plus these two, or you could just subtract 1 minus this. That would be another way to get there. But anyway you get there, 0.42 plus 0.9, 0.51, 1 minus 0.49, 0.51. So the frequency of the dominant phenotype is 0.51. So that's how you solve Hardy Weinberg related problems. Do it this way. You'll get it right every time. If you try and solve it using binomial factoring and things like that, I mean, hey, if you can do that, do it. Go ahead. But I can't do it that way. So I always taught my students to do cross multiplication tables. We're going to now look at an illustrative example. Sickle cell disease is caused by a recessive allele in the gene for hemoglobin. I talked about this extensively in my reviews of Unit 6, which you can find on the Science Music Videos channel. So this mutation, this recessive allele, can significantly decrease the quality of life and lifespan, yet the allele is in high frequency in certain populations explained. So this is a map of Africa, and it's showing the percent of the population that has the sickle cell allele. And notice how high it is in areas like this and this over here, and even in some areas of India. So what is going on? Well, sickle cell disease only occurs in children who are recessive for the HBS allele. So you have to have little s to actually get the full-blown disease. That full-blown disease is a very, very difficult disease. I talk about that again in Unit 6. It causes pain crises as these red blood cells sickle. They get caught in tissues, so on and so forth. If you're a heterozygote, if you have the big S, little s genotype, then it changes the chemistry of your blood a little bit. There's a small amount of sickling, but not enough to generate the pain crises and tissue damage experienced in people who are homozygous, little s, little s. But that makes the red blood cells in those heterozygotes, big S, little s, a hostile environment for a parasite called Plasmodium that causes malaria. In other words, if you're a heterozygote, you have a kind of resistance against malaria, which is a deadly disease that kills hundreds of millions of people in areas where malaria is common. It causes these recurrent fevers. If you can imagine this, the parasite is so small that it can live inside of red blood cells. And it goes through a cycle where the parasite bursts out of the red blood cells, and that causes debilitating fever. Usually that has been controlled, or it's been attempted to be controlled by wiping out mosquitoes, but because of evolution, that can't be done. And we'll talk about that in a later episode of this evolution review. So the result is heterozygotes have resistance to malaria, and that has led to natural selection that has increased the frequency of the sickling allele in populations that live in malaria-prone areas. Well, benefit, more heterozygotes survive. Cost, more homozygotes die, but also more homozygous normals. In other words, big S, that genotype, that's also selected against because of malaria. So we have selection against being homozygous recessive because of sickle cell anemia. There's selection against being homozygous dominant, big S, and there's selection for little s. This phenomenon is called heterozygote advantage. It explains the high frequency of an allele that's harmful in homozygotes. I have a song that I'd like to play a little bit for you. It's called The Ballad of the Peppered Moth, and let's just listen to a little bit of it. Meet the peppered moths of the English woods. The forest of Manchester is its neighborhood. It's gone from white to black and back through natural selection from bird attacks. Natural selection starts with inherited variation, followed by selection of favored traits within a population. If selective pressures consistently apply in the same direction, the beneficial traits will become the norm. It's how adaptations form. Super fun. I want to let you know about a couple of resources that you should know about. First of all is that if you go to learn-biology.com, you go to the courses menu, and then you go to AP Biology version 2, and then you'll make your way to the evolution main menu. I've got tutorials about every single topic. Those tutorials include readings, they include interaction. Do these tutorials, you won't believe how much you'll learn. Here's the one about sexual selection. It starts with a little game, and then there's reading, and then there's some identification that you have to do. There's a little bit more reading, there's a quiz, and there's flashcards. Think about how much you would know about this topic if you learned like that. There's fantastic AP Bio reviews. So here's our AP Bio FRQ review. And for example, if you went there and you wanted to do a quiz and focus on the FRQs, then you could just select the ones in Unit 7. And in fact, you could just review the stuff that we did. You just select natural selection, population genetics, and Hardy Weinberg, and you'd have a couple of problems. You'd have an FRQ that you could respond to, and then you could see the sample answer. So this is a way that you can really hone your skills. And then finally, I want to tell you about that checklist that I was mentioning at the beginning of the video. If you go to apbiosuccess.com/checklist, you can sign up to download this fantastic checklist. The checklist is going to really be an important edge that will help you study, and you can use it in conjunction with learn-biology.com. So I want to thank you. I want to thank you so much for joining me. We have a schedule of live review sessions at learn-biology.com/live sessions. And, oh, that's a problem because I haven't made it a public page yet. Well, one of the several things that weren't perfect about this, but anyhow, we'll make it better. And I really appreciate your joining me. I'll fix this link. Just go to learn-biology.com/live sessions, and you'll be able to experience all of these review sessions. Next one is either Sunday or Monday, but I am really honored that you chose to spend this time with me. Good luck learning biology, and you'll learn it best if you learn it on learn-biology.com. Thank you so much for joining me.